The overall goal of the following experiment is to discover and identify long-range associated DNA partners. This is achieved by fixation, cell lysis, and digestion of DNA by the first restriction enzyme, followed by ligation to create a chimeric DNA composed of segments from the two associated genes. Next, linkers are added to the chimeric DNA after the second restriction digestion to facilitate its amplification. PCR amplification is then performed using linker and bait DNA-specific primers in order to obtain sufficient amounts of the associated DNA fragments. Results are obtained that show the long-range associated DNA partners with the bait DNA based on sequence analysis of the amplified DNA fragments.
The main advantage of this technique over existing methods like chromosome conformation capture or 3C is that ACT assay can be used to identify a no long-range associated DNA partner effectively, whereas 3C allows for verification of only previously suspected interactions.
To begin this protocol, culture human cells in an incubator supplied with 5% carbon dioxide at 37 degrees Celsius until they are 80 to 90% confluent. Human promyelocytic leukemia cells are used for this demonstration. Collect the cells by pouring them into a 50 milliliter Nunk tube and centrifuge at 1200 RPM for 15 minutes.